Hello students, welcome to EPG Pathshala. I am Dr. Zeenat from Department of Pharmaceutics, Faculty of Pharmacy, Jamia Hamdard. Today we are going to talk on the module Nasal Drug Delivery System, Formulation Aspects and the various transport mechanism. This falls under the paper NDTS 1. My dear students, let us see what we are going to learn in this particular module. Our main focus would be getting introduced to the various formulation factors which will affect the nasal drug bioavailability. Then we will spend appreciable time in understanding the transport route and mechanisms, because once we are sure that how this drug will penetrate through the mucosal membrane, then only we will be able to find out how it will work therapeutically. So we'll spend a lot of time on understanding the transport routes. Then there would be a very fine section which will let us understand the role of the transporters and the efflux systems, which will ultimately affect the nasal drug bioavailability. So my dear students, as we will start discussing about the various formulation aspects, I think again not to reiterate, the formulation parameters would be taken into considerations. Whenever we are talking about a route of administration, we need to talk about the various aspects. We have already discussed in other classes that there are various physiological factors which will affect the drug bioavailability. But when we talk about the formulation, the solubility of the drug, the lipophilicity, the partition coefficient, and many more factors will play a very important role in the actual drug being made available at any particular route, and nasal route is no exception. So we would go into talking about the various formulation factors which will actually affect the nasal bioavailability. We have summed up it in different categories. So we will take up the first as the factors which are directly related to the drug itself. So the first factor which probably has to be discussed is the chemical form of a drug. Chemical form of a drug has a very important effect on the nasal absorption. The salt form or the ester form of a drug can have a significant effect on the pattern of nasal absorption. The second one is your lipophilicity. When we talk of the lipophilicity, the increase in lipophilicity of the drug results in enhanced permeation through the nasal mucosa. The nasal mucosa per se is lipophilic in nature, and the lipid layer which is present onto that mucosa will probably play an important role in the barrier functions of these membranes. So if a drug is lipophilic in nature, it would be probably attracted more towards the membrane and enhance the permeation. It has been found by many researchers that the lipophilic compounds such as your alpranolol and propranolol were very well absorbed from the nasal mucosa. The hydrophilic drug, on the other hand, like your the same category, that is antihypertensive, but we have a drug by the name metoprolol which is absolutely hydrophilic in nature, and this, when was given through the nasal mucosa, had a very reduced absorption. The lipophilic compounds cross the biological membranes through the transcellular route due to their ability to partition into the lipid bilayer of the cell membrane. Further, they may diffuse and traverse the cell in the cell cytoplasm. In continuation of studying the various drug aspects which will be of importance in the nasal drug delivery, we come to the next important point, that is polymorphism. The purity of the drugs as well as the polymorphic stability are very important characteristics for the nasal powders and suspensions. It was found that polymorphism affects the rate of dissolution and solubility of the drugs, which ultimately affects the absorption pattern of the drug through the biological membranes. The next important parameter is the solubility and the dissolution rate. For powders and suspensions, solubility and the dissolution rates of drug are important factors for determining the nasal absorption. The particles need to be dissolved in the nasal cavity prior to the drug absorption. If a drug remains in the particle form, no absorption takes place. Molecular weight is the next important parameter. The water-soluble drugs maintain an inverse relationship between the weight and the drug permeation, while a direct relationship exists for the lipophilic drugs. Drugs with molecular weight less than 300 Daltons are not significantly influenced by the physicochemical properties of the drug, but the drugs with molecular weight greater than 300 Daltons are highly sensitive for permeation rate across the nasal mucosa. Partition coefficient and the pKa are another important set of parameters which will affect the drug delivery. The unionized drugs are absorbed better in the nasal cavity as compared with the ionized species. A study was conducted to determine quantitative relationship between the physicochemical properties of the drugs and their nasal absorption using two model drugs. The drugs which were chosen as the model drugs were diltiazem hydrochloride and the common paradrug paracetamol. The results obtained from the experiment showed a quantitative relationship between the partition coefficient and the nasal absorption constant. The nasal absorption of weak electrolytes such as the aminopyrine and salicylic acid was found to be dependent on its degree of ionization. The absorption rate of aminopyrine was found to be increased with increase in pH, but some kind of variations were observed with salicylic acid. In continuation, what we have just understood about the factors which are primarily related to the drug, we also have a group of physicochemical properties of the formulation which will definitely have a big impact on the drug formulation and finally impact on its delivery to through that particular route. So the first one is your viscosity. The contact time between the drug and the nasal mucosa increases when the viscosity of the formulation is high. It will ultimately lead to increased time for penetration. Something which is viscous, it will probably be staying there for a longer duration of time, and by default it will be there having an increased time for permeation. So this is an important parameter which is to be borne in mind. In addition to it, highly viscous formulations affects the regular function of the nasal mucosa. Of course, then probably I think again we can very well appreciate that if there is a lot of secretions in that particular site of action, already there is a large pool of secretions there. On top of that, you are having the drug which is present there and that is again viscous. So the viscosity of the drug probably will play a very important role. Then we have the next factor, that is your osmolarity. The effect of osmolarity on the absorption of various agents has been studied by many researchers. It was observed that the absorption of serotonin reached a maximum level in the nasal mucosa at a sodium concentration of 0.462 molars. The observed effect, if we try to understand, was probably due to the shrinkage of the nasal epithelium leading to the enhanced permeation of the compound. So probably we are having a osmolarity of a particular tune, so it might affect the nasal mucosa, maybe shrinkage is happening, some other physiological changes are happening, and this might enhance the permeation of the compound. This was then very probably most in detail studied with the compound which is an osmoregulatory agent like sorbitol, and it was further confirmed, yes, it probably plays an important role. Hence, ideally, what we should do as a formulation scientist is the isotonic solutions are preferred for the nasal administration where the membrane would not be compromised. Then we have another important factor, namely the pH and mucosal irritation. Let us talk about them together. Ideally, the pH of the intranasal formulation must be between 4.5 to 6.5. This is very ideally suited if we want that should be no nasal irritation. If I am sticking to this pH of this range, then probably I will also be able to get an enhanced drug permeation and prevention of the bacterial growth in the nasal epithelium, because this is a most ideal pH where these problems can be easily circumvented. My dear students, we would now continue to understand what are the other factors which would play an important role in the designing of the nasal drug delivery systems. There is an important set of factors which are associated with the dosage form. Drugs administered in the nasal cavity are either formulated as nasal drops or nasal sprays. These are the two commonest type of dosage forms which we see in the market and common usage. The nasal drops deposit in the nasal cavity as a film layer of drug solution, and the nasal sprays deposit an aerosol of droplets, particles, or particles suspended in the droplets. In case of aerosol formulations, the deposition of aerosol is the primary step in the nasal cavity, followed by the dissolution of the solid particles. The rate and extent of deposition of an aerosol, for example, from the nasal spray depends upon the following factors. We'll just try to understand each factor individually. The first one is the particle charge. The particle charge, it depends on the drug, formulation excipients, or the method of aerosolization. The next one is your aerodynamic diameter of the particle, which is a function of droplet size, shape, and density. And finally, it is the velocity of movement of particles, which depends upon the pattern of respiration. The droplets in the size range between 5 to 10 micrometers deposit in the nasal passages. The extent and site of particle deposition can be complicated by the increase in the size of particles due to water condensation, which may be due to humidity change upon entering the nasal cavity. Deposition mechanisms in the nose usually include inertial impaction, sedimentation, diffusion, interception, and electrostatic attraction. The nasal gels used in the nasal cavity are high viscosity solutions or suspensions. The various advantages offered by the nasal gel include the reduction of the post-nasal drip due to high viscosity, decrease in taste impact due to reduced swallowing, reduction of leakage from the formulation, decrease in irritation rate by using the soothing excipients, and targeted delivery to nasal mucosa for better absorption. Nasal powders are also administered as dosage forms in the nasal cavity due to the lack of use of preservatives and superior stability of the formulation. The suitability of the use of powder formulation is largely dependent on the solubility, particle size, aerodynamic properties, and nasal irritancy property of the active ingredient or drug and the excipients used. The irritation of nasal mucosa is avoided due to the local application of drug, which is another advantage of this system. But nasal mucosa irritancy and metered dose delivery are some of the major challenges for the formulation scientists and device manufacturers, which needs to be resolved. The absorption enhancers act by one of the following mechanisms. Either these will inhibit the enzymatic activity or they will reduce the mucus viscosity. Then they can further decrease the mucociliary clearance, and sometimes they also work to open the tight cellular junctions. Generally, the absorption enhancers are classified as physical enhancers and chemical enhancers. Chemical enhancers act by destroying the nasal mucosa, while the physical enhancers affect the nasal clearance reversibly by forming a gel. Some of the examples which can be used for chemical enhancers include the chelating agents, fatty acids, bile acid salts, surfactants, as well as quite a number of preservatives. After understanding the various factors which will affect the formulation design of the nasal drug delivery system, let us go and take an insight into the transport routes and mechanisms which these drugs will traverse or face while being delivered. Drugs which are applied topically to the nasal conjunctiva enters the nose through the nasolacrimal duct, which is located just beneath the anterior end of the inferior turbinate. The other conducting airways permits the total passage of respired air with a minimum resistance to the inspired airflow. The nasal cavity, as already been discussed earlier, has a unique architecture. The whole of the nasal cavity is bifurcated into two equal halves by the nasal septum. Due to this bifurcation, the nasal airways present much resistance to the airflow. Each half of the nasal cavity consists of many folds, which in addition to the provided resistance allows for an appropriate intimate contact between the mucosal surface and the airstream. The olfactory region remains free of inspired air. The nasal region is also supplied with highly vascularized erectile tissue, where most of the intranasally administered formulations such as nasal drops and nasal sprays reach. Due to the nature of circulatory and mucociliary systems, as well as the close contact between the mucosal surfaces and narrow airstream, the stabilization of water vapor content and temperature occurs. A number of goblet cells and submucosal glands comprise the mucociliary system, which provide mucus and cilia that helps in the proper transport of fluids to nasopharynx. The circulatory and secretory mechanisms are susceptible to a number of influences. For example, the capacity of the nasal cavity to transmit topically administered drug to the systemic circulation is effectively reduced if a certain factor produces certain limit of significant vasoconstriction. Certain factors influence the delivery of drug from formulations which are administered into the nose, drop in the form of liquid or spray. The constriction present in the anterior area of the nose is known as the nasal valve. The inspired air passes through this point. High linear velocity and change direction of the inspired air are two major characteristics which result in deposition of most aerosols in the anterior region of the nose. Larger particles are usually administered and do not absorb in the nasal cavity into the systemic circulation because they lie behind anteriorly in front of the exchange region of the nose. Insoluble particles mainly deposit in the main passage of the nasal cavity and are likely to be carried backward by the mucociliary system and dispatched to the stomach. If a drug is administered in the form of a vapor or in a soluble form, it may readily pass through the surface secretions and into the systemic circulations. Nasal polyposis resulting from nasal obstruction leads to reduction in the capacity of the nasal absorption of the drugs. Additionally, pathological conditions such as atrophic rhinitis or severe vasomotor rhinitis also reduce the usefulness of the nose to absorb an administered drug. Intravenous route of administration serves as the reference standard for establishing the basis of bioavailability out of all other routes of administration. Intranasal route of administration presents an attractive route of administration for drugs of the categories like hormones, peptides, vaccines, and many other peptide-based drugs. It presents a distinct advantage for drugs which undergo extensive hepatic first-pass elimination, gut wall metabolism, and destruction in the GIT fluids. The efficiency of particle removal from the inhaled air stream is dependent upon the number of factors which include the aerodynamic diameter of the inhaled particles. Particles greater than size of 10 micrometers are generally filtered out by the vibrissae at the nostrils. Smaller particles in the range of 5 to 10 micrometers tend to deposit in the nasal passages and are subsequently cleared by the process of mucociliary clearance. Particles less than 2 micrometers in size are not normally filtered out and may enter the lungs. My dear students, it has been a few decades that the role of various transporters, particularly of the category of ABC transporters like PGPs, have been elucidated in the drug delivery across the various membranes. The nasal epithelium is also not untouched by the effect of this transporter. The PGP transporters or the P-glycoprotein transporters have been found to be playing an important role in the transport of drugs across the nasal epithelium. It has been observed that the drug absorption can be reduced due to the efflux transporters, namely the PGPs. Let us try to find out what are these PGPs. P-glycoproteins, acronymed as PGPs, are the large glycosylated membrane proteins which are found primarily in the apical membrane of the epithelial cells of the small intestine and in various other tissues throughout the body. The two PGP genes which code for MDR1 and MDR3 proteins are present in humans, whereas rodents have three coding specifically for MDR1A, MDR1B, and MDR2. The P-glycoproteins have been found to have the ability to probably transport a broad range of drugs with no common structural characteristics. So you cannot say that only a particular category of the drugs will be affected by the presence of the PGPs. The drugs can range in size from 250 daltons like cimetidine to about 1900 daltons like gramicidine. The drugs that are effectively transported through the nasal mucosa are primarily basic or uncharged in nature. All PGP substrates which are discovered are usually amphiphilic in nature. MDR1 and the multi-drug resistance associated protein MDP1 are present in the human nasal respiratory mucosa, wherein it has the ability to transport a large variety of hydrophobic and amphiphilic substrates and is known to be effective in cell detoxifications of xenobiotics. MDR1 is also known to transport a range of peptides such as peptide growth factors. These two above mentioned transporter proteins have been found in olfactory receptor neurons in tadpoles as well. The levels of MDR1 in the respiratory epithelium in humans are lower by approximately 250-fold than the levels in the liver. The expression of PGP is also enhanced by the administration of topical steroids in the nasal cavity. It has also been found that the PGP attenuated the transport of a wide variety of drugs which were substrates for PGP to brain tissue following intranasal administration. Rifampin, which is known to be a PGP efflux inhibitor, increases the uptake of the drug in the brain when given in a combination. The specific transport of PGP substrates such as chlorcyclizine and chlorpheniramine was studied in vitro using the excised bovine olfactory mucosa. The observed effects supported the fact that the transport of the drug was greater from the serosal to the mucosal side of the tissue than from the mucosal to the serosal side. In addition, the flux was also found to be concentration dependent and is saturation limited. P-glycoprotein was predominantly found in the apical regions of the ciliated epithelial cells and in the submucosal vessels. PGP inhibitors such as verapamil and MRP1 inhibitor such as quinidine inhibited the PGP efflux. The conclusion of the above study was found to be that the PGP which are found in the olfactory epithelium played a major role in preventing the drugs from crossing the nasal membrane. It is also known that due to the presence of efflux transporters in the olfactory epithelium, drugs such as chlorcyclizine and chlorpheniramine did not reach the CSF in rats. These efflux transporters also act as a potential barrier in the transport mechanism in subepithelial lymphatics and vascular endothelium. Histological and immunohistochemical analysis of excised bovine tissues also supported the presence of PGP efflux transporters in both the respiratory epithelium as well as in the olfactory epithelium. The concentration of PGP was also found to be much higher in the olfactory region than in the respiratory region. Another example is of the drug etoposide. The efflux of the drug etoposide by PGP was found to be significant, and the efflux of the drug was decreased by the inhibition with 2,4-DNP and quinidine. The influence of the efflux PGP inhibitor rifampin on the transport of the drug verapamil from the nasal cavity as well as from the systemic circulation into the brain of mice was also studied in detail. In this study, rifampin was administered both nasally and intravenously. Irrespective of the administration route, the drug inhibited the efflux of verapamil. The highest inhibitory effect was observed when both the drug and the inhibitor were given nasally, resulting in approximately 100% inhibition. The inhibition effect of rifampin was found to be most efficient after intranasal administration, due to which a much lower concentration was required after intravenous administration. But the extent of systemic distribution of the drug or inhibitor after nasal administration could not be studied in detail. For nasally administered rifampin, the concentration of the drug in the brain was located in the distributed pattern than after systemic administration, in which the brain distribution was more uniform. Pharmacokinetic modeling was also implemented to investigate the pharmacokinetics of uptake and the distribution in the brain of model drugs that are specific substrates for PGP. In this model, PGP-mediated efflux was found to be larger from the nasal epithelium than from the olfactory epithelium, mainly due to the larger surface area of the nasal mucosa. The study supported the fact that PGP is functional at the olfactory epithelium and attenuates the brain uptake of drugs after nasal administration. It was also demonstrated that PGP operates through the brain to efflux substrates during rostral to caudal distribution. Hence, it can be concluded that the existence of transporter systems in the nasal cavity and their effect on absorption of drugs from the nasal mucosa into the systemic circulation and on transport into the CNS is a novel as well as innovative area of research. The future developments to implement the role of these systems in achieving therapeutically relevant concentrations of drug in the CNS may have a significant impact on the development of drugs formulated for intranasal delivery. So students, this has brought us to the last part of the module today. When we are talking about, we have in detail discussed about the various factors which are going to ultimately affect the nasal drug delivery. So let us try to summarize what we have learned through this module. The first thing which we have been able to identify is that the nasal drug delivery and the bioavailability is definitely affected by the properties of the drug, such as the molecular weight, the solubility, the lipophilicity. We had quite a good number of examples to reiterate this point. Then probably the second important take-home message is that it is also governed by the formulation factors such as the viscosity, the pH, the osmolarity, as well as the various other dosage form factors. We in detail studied about a very novel concept where we had probably understood that the P glycoproteins, they play a major role in the transport of drugs across the nasal epithelium. We concluded that the drug absorption can be greatly reduced due to the efflux transporters such as the PGPs. Presence of PGP also affects the transport of drug from the nasal cavity to the CNS. So what we can probably conclude is that we are at the verge of discovering futuristic drug delivery systems which can be given through the nasal cavity, but which can definitely exploit, modulate the transport system and these systems are absolutely under investigation. If they can be put into right perspective, they can definitely facilitate delivery of the drug via the nasal route for systemic circulation as well as through the blood-brain barrier.